Clinicians can also rely on more modern techniques, such as molecular diagnoses, to diagnose an infection. When I say molecular diagnosis, I'm talking about things like DNA or protein detection. When we're talking about DNA or RNA, so any of the nucleic acids, if we're trying to detect them, often that will require amplification of the genetic material prior to testing. And what we're talking about here is PCR and its derivatives like reverse transcriptase PCR, qRT-PCR, things like that. So this is what PCR looks like. Again, you should all be familiar with PCR. If you've taken genetics, you should have taken genetics, cell biology, things like that. You've talked about PCR, you know what it is. Once you have amplified DNA or a section of DNA, a really common tool that sounds a little bit old school, but actually works pretty well, is to use restriction enzymes to digest that amplified DNA. And many microorganisms can actually be distinguished by the pattern generated when their DNA is digested with particular restriction enzymes. You then run that digest out on an agarose gel, and you can visualize the different patterns. And we call this type of assay a restriction fragment length polymorphism, or a RFLP. I like saying RFLP. So RFLP. In this pattern here, what we can see is that 1, 2, and 3 have the same digest pattern. So that's going to be the same microorganism. 4 and 5 are the same as one another, but different from 1, 2, and 3. And then 6 is out here kind of all by itself, different from everybody. So this would be an example of 3 different microorganisms. And I don't even mean they have to be different species. This could be 3 different strains of the same species could still give you these different digest patterns. So this is a quick and easy way to identify an infection. This is also commonly used in tracking outbreaks. So where did the disease come from? Did it come from, like in the food chain, right? So if we talk about spinach that has E. coli, O157H7, did it come from the packaging facility? Did it come from the transport? Did it come from the field? Like, where did it come from? These types of assays are really good for that. And of course, there's DNA sequencing, and maybe this is the wave of the future. And in your reflection, you can maybe hit on this. Do you think DNA sequencing is the wave of the future based on what you know about various DNA sequencing technologies? There's whole genome sequencing, which allows you to look at every single nucleotide found in that organism. There's 16S rRNA sequencing that can allow you to maybe identify particular species as members of a microbiome. So DNA sequencing, is it the wave of the future? Is it as rapid as a RFLP? Is there a way to make it more rapid? Is there a rapid type of DNA sequencing? Is it fast enough to diagnose a patient? These are all things that you have to think about as a clinician, because for your patients who have an infection, this really could be a matter of life and death. In molecular diagnoses, we can also look at proteins. And when I talk about proteins, they could be proteins from a microbe, or they could be proteins like antibodies that your body has produced in response to an infection. There are a number of different tools for protein detection that can be used, including mass spec, SDS-PAGE, Western blotting, etc. So here is MADLI-TOF. It's a type of mass spec. The chemistry behind this is even more like beyond what I'm interested in. This is not my research interest, but this is something that can identify very particular proteins. And you can see an image about how mass spec works here. There are other types of immunoassays listed in this table. As we talk about various infections, we'll talk about the different diagnostics that are common for those infections. But some of the more common techniques that you might see in a clinical laboratory setting would be something like an ELISA, which can allow you to quantify an antigen or an antibody produced by the host. Western blots can also, again, detect antibodies produced by the host or antigens found within. There's variations of ELISA, complement fixation, hemagglutination. So there are a number of different ways that scientists use to diagnose various infections. And you can see here, some of those infections are listed here. So you could do a Western blot for a patient you suspect might have HIV, complement fixation for fungi or viruses, hemagglutination for various influenza strains, latex agglutination for fungal antigens, streptococcal antigens. A lot of times when we're talking about using proteins, either looking for antigens or for measuring the host response by looking at antibodies, we are talking about viral infections. And I want you to think about in your reflection, why would we depend on these types of assays, these molecular assays, more for viral infection than for something like bacterial infection? So in your reflection, I want you to think about why that would be. As we talk about those types of infections, we'll talk about why. So we'll get there, but I want you to think about it now. When I use the term antibody, what I mean is this is a protein that is produced by the body, and it is going to be specific for a various antigen. There are two types of antibodies. There's monoclonal, which are large quantities of antibodies that only bind one very particular antigen. These are generally not how your body responds to infection. Your body doesn't make only one antibody to attack one antigen of whatever infection you have. These are most often made in a clinical setting. And they're very specific. What if the antibody is actually effective against an intracellular antigen, like an antigen inside of a bacterial cell? How is that antibody going to get there to bind to the bacterial cell to trigger the immune response? It can't. So what happens in our body is we generate what are called polyclonal antibodies. They're specific for different epitopes of the same antigen. So let's say we have a staph infection. Those are super, super common in the skin. What happens is cells of your immune system, and we'll talk about the immune system later this week and into early next week, cells of the immune system will come in and phagocytize those staphylococcal cells that got in your body. They'll chop them up, so you'll have presentation of pieces of staph to the immune system. And they could be pieces from the outside, pieces from the inside, pieces from the front, pieces from the back, whatever. And so you get this huge population of antibodies that recognizes lots of different parts of staph. And so that's really great because then, maybe you do have some antibodies that only recognize the inside, but you have lots of antibodies that will also recognize the outside. So you get the chance for your immune system to respond. The downside is, and we'll again talk about this as we talk about various pathogens, sometimes pathogens have particular antigens that look a lot like our proteins. And so you can get an issue where your antibodies may bind to your own proteins instead of just the bacterial proteins or the viral proteins or the fungal proteins. So you might get those non-specific interactions. I'm not going to explain all of these different tools, and because I'm not explaining them in a lot of detail, I'm not going to expect you to know a ton about them either. But I do recommend you read these sections in the textbook or you look up these different types of assays because they may be very important for you who are going into clinical fields. So you have a number of assays you can do in a laboratory setting that are antigen-antibody interactions. So generally with these, you are probably taking host serum. So you're taking blood from your patient and isolating out the fraction that would contain antibodies, and you're testing it against known antigens. But sometimes you may have antibodies that are specific, like commercially produced antibodies specific for a particular antigen. and you're looking for the patient to have produced the antigen. That's how something like a COVID test would work. You can have immunofluorescence, where you have, here they're showing you an antiviral antibody, and it's been tagged with a fluorescent probe. And so if it finds a cell that's infected, it will bind, and you can visualize that on a microscope. So here they're showing herpes simplex virus infected nerve cells in the brain from a patient who has herpes encephalitis. You can use antigen markers to cause fluorescence in cells, and there's a machine called a flow cytometer, or a fluorescence activated cell sorter, a fax. Not fax like FAX that used to be used to send papers, but fax, FACS, that can actually sort cells based on fluorescence tags. So these, if they have CD3 versus CD4, based on cell size. So flow cytometers are very cool technologies as well. And then I mentioned ELISAs. These are very, very common techniques in immunology labs. And you can run them a couple of different ways. Again, if you know, like let's say you have an antigen and you want to test whether or not the patient has made antibodies against that antigen, you can coat a plate with the antigen, take the patient's serum, and look for a reaction. Or let's say you suspect a patient may have an infection, but what we'll talk about in immunology is that it can take time for the antibodies to develop, like a week or more. So you want to see, does the patient have the antigens? So then you take antibodies that are specific for the antigen, you add the patient sample that may have the antigen in there, and you look for a reaction. So this is sort of how a COVID-19 test would work, and you've probably taken some of those. So what happens is there are antibodies in the little COVID tests. They call them a lateral flow assay because it runs horizontally instead of vertically. And so there's little antibodies in there, and as you swab your nose, you mix it with the liquid, you put it on the sample, and you let it run. And what happens is there's a control line. So it looks for, well, are there human proteins in here? Yes, perfect. That's your control line. But then it's looking for COVID proteins. So are there COVID-19 proteins in your sample as well? And that's when the test line will become positive, because you'll have a color change when you have this type of antibody antigen interaction. So it's looking for your nasal passages specifically to have COVID-19 antigens. And so this type of assay where you're looking for the antigens can be beneficial because it can allow you to detect the presence of the infection prior to the immune response to the infection. So a patient will be positive for having antigens before they're positive for having antibodies. Western blot is a variation on an ELISA. These are not used as commonly as an ELISA or even one of those lateral flow assays, but they're still used to diagnose infections like Lyme disease and HIV. Again, here's a summary of different types of serological assays that again will involve host serum. And the idea is that you can try to ID the infection. You can evaluate the course of the infection, so you can look for the antibodies that can tell you it's been a longer infection than before. You can determine if it's primary or secondary. What we'll talk about when we talk more about antibodies in the immunology section is how you have a switch from IgM to IgG. And so looking at the ratio can tell you the course of the infection or if it's a secondary infection. We report the antibody concentration as a titer. And during the primary infection, once the body starts producing antibodies to the infection, we call that seroconversion. And then there's a term for the secondary response. I always mess this up. Anamnestic, which is like the memory response to the second, third, fourth, whatever time you've had that infection. So again, serology is generally used to diagnose viral infection, and as part of your reflection, I want you to think about why. These are some of the viruses listed here that can be diagnosed by serology, by looking at, when we say serology, what we mean is looking at your serum, looking at your antibodies, and trying to diagnose the infection. Sometimes you can't use the antigens. So for example, for HIV infection, we don't look for the HIV antigen because usually it's found at very low levels. So we look for the presence of the antibody instead. And that's why immediately after potential exposure to HIV, you wouldn't want to test because it would be negative. You want to wait until seroconversion might have happened. Then you can test for the presence of antibodies. Another example of using serology is maybe some of you are interested in reproductive medicine. And so you would want to test patients who are trying to get pregnant. You would want to test them for the presence of rubella titers. Rubella is part of the MMR vaccine, so it's the R part of the MMR. And rubella infection during pregnancy can cause really significant issues for the fetus, including all the way up to stillbirth. So while the majority of patients are vaccinated as children, every once in a while, either you have a patient who wasn't vaccinated or you have a patient whose immune response has waned. And so you can test their titers for rubella and see if they're still protected. And if not, you can have them get another MMR shot so that they would be protected during their pregnancy. So there are lots of reasons you might want to measure a patient's titer, not just to determine whether or not they've had an infection, but to determine whether or not they might be protected from future infection.